Okay, so welcome to this video. So today we are going to talk about a very interesting topic of the chromatographic analysis, and that is none other than how one can investigate the extraneous peak observed during the chromatography run into a test preparation. So in case if you are conducting a routine analysis for your test sample, or maybe a stability analysis, and you have found something a very unique peak which was not observed into the earlier batches or during the earlier stability station, then you have actually found the extraneous peak. So let us begin with the presentation now. And as a part of today's presentation, we are going to discuss about these five important points. What is the definition of extraneous peak? What are expected sources of extraneous peaks? How to conduct the investigation for extraneous peak? How to identify and qualify the extraneous peak? And what is the fate of the batch now? So let us begin with the very first point, and that is the definition of extraneous peak. And here it is. So the peak excluding diluent, placebo, API, or salt form of the API is called as the extraneous peak. Now, what is the salt form? For example, if you have a enalapril malate as your API, so you are expected to have two peaks onto the chromatogram of the API or the standard. One belongs to enalapril base and the second will belongs to a malic acid. So that is called as the salt form. Peak excluding degradants observed during forced degradation studies. So if this peak is not observed during the degradation studies that you have conducted during method validation, then that peak can also be called as the extraneous peak. And the third is the peak excluding residual solvents. So in case if your API is manufactured by using some of the residual solvents like methanol, tetrahydrofuran, and if that peak is not belongs to the residual solvents, then you can say that this is the extraneous peak. So what are the expected sources for the extraneous peak, and here are they. So the cross contamination during manufacturing, you are manufacturing the batches and you are manufacturing, let us say, different products, paracetamol followed by metformin. So in case if the paracetamol is not cleaned out, from the surface of the manufacturing equipment, then the traces of paracetamol can get carried forward into the metformin, and which can become the source of cross contamination. So the cleaning of equipment can become the source of cross contamination. The second can be a cross contamination during analysis. So you are using the used glassware, and if the glasswares are not cleaned properly, probably they can they can become the source of the extraneous peak. So the leftover residues can become the source of your extraneous peak even during the analysis, and it can be your pipettes, it can be your volumetric flask, it can be your HPLC vials. Those can become the source of cross contamination. Leachables from container closure system. So in case if you're not observed these leachables earlier, but for some reason maybe during stability now you are finding some leachables coming into your product. So these leachables can also become the source of your extraneous peaks. Excipients and container closure system interaction. So in case if your container closure system is also interacting with the excipients and forming some new impurities, then this kind of impurities can become the source of extraneous peak. So these are the four important sources for the extraneous peak. The third important point is now, which is very important, it is the investigation. And we are going to talk about 13 different points as a part of investigation. So how one can investigate the finding of extraneous peak, and let us begin with the point number one, that is the first and foremost, you need to very much confirm that this peak is really an extraneous peak or not. So how you can do that? Just looking at the forced degradation data. So you may having the oxidation, thermal degradation, alkali or acid hydrolysis, and based on to those chromatogram you can understand, okay, now this is the peak which is not observed in any of the forced degradation, and hence this is the confirmation for extraneous peak. Placebo chromatogram, so confirm the placebo chromatograms or even the diluent chromatograms for one more time, and then understand, okay, now this is the exact extraneous peak I found. So confirmation is the first step of the investigation. Then study the past history of this product, whether this kind of peaks were already observed into the previous stability station or into the another batches that has been released or manufactured into the same manufacturing unit, or maybe into the another manufacturing unit. So understand the history of this peak. If you have come across no such kind of situation, then you can think of, the planning the few of the experiments to understand the laboratory error. So before you go on to the product related issues, first understand whether there is any possible lab error for the finding of this extraneous peak. And the first you can think of re-injecting the same vial solution once again to identify whether the source is not belonging to the instrument or the HPLC or the gas chromatography. So to rule out instrument related artifact, you have to re-inject same vial solution one more time. Second can be the fill the test solution into new vial. Again use the same available test solution, but by using the new clean HPLC or GC vial, and then inject to identify whether the vial was not the culprit, whether the extraneous peak was because of the vial contamination. To rule out the vial contamination, re-inject the same test solution available, but by using the another fresh or a new HPLC vial or a new JC vial with the suitable septa. To identify whether the origin of the extraneous peak is not because of the filtration, refilter the test solution and then understand whether now you are getting the same peak once again. Also understand whether you have used the suitable filter paper, whether the PVDF was mentioned into the test procedure or the same was used or not. So identify the root cause. Is it because of the filter or filtration? Then the also identify whether the possible extraneous peak is from the glasswares used. So you can think of, if there are multiple dilutions to be done, right? You can think of using the same stock solution, but by using the fresh, well-dried glasswares and pipettes, so that you can identify whether the extraneous peak is due to the glassware contamination or not. The point number 7 is extend the chromatographic run to identify whether this impurity is the strongly eluting impurities, and whatever you are finding into the chromatogram is actually the carryover of the late eluting impurities. So extending a runtime is also a good thought process in identifying the extraneous peaks origin. Then inject solution prepared out of primary pack. So now you have identified that origin is not because of your glassware contamination, the vial contamination. You need to think starting on to the source as a primary packaging material. So if the primary packaging material, because this is the packaging material which comes in a direct contact with your product, and if there are some leachables coming out of the packaging materials, you need to identify those leachables. So prepare the test solution by using the primary packaging materials, like let us say alu pack. So prepare the solution by using alu pack, alu foil, and then identify whether you are finding the similar extraneous peak into the chromatographic run. Inject solution prepared out of printing labels. It has been found that the some of the printing inks are also can become the source of extraneous peak. Because this is also a very important point, and the printing label also a part of your primary packaging material. So identifying or investigating the printing material can be a good idea of investigating the extraneous peak. Inject solution prepared out of a previous product to identify source as a ineffective cleaning. So in case the paracetamol followed by metformin, right? So paracetamol was your previous product. So inject the paracetamol API or standard and identify the retention time of paracetamol. So you can easily understand whether if the paracetamol was the previous product, was that the reason for the extraneous peak. The point number 11 is inject solution prepared out of parallel running product to identify cross contamination during manufacturing. So if you have a multi-product manufacturing facility, it can be API or finished product. Understand whether those parallel running products can become the source of cross contamination. So just inject the APIs of those products and you will come to know the source of extraneous peak. Inject solvents used in manufacturing to identify source as a residual solvent. See, some of the residual solvent like benzyl alcohol, dimethyl sulfoxide can also give a very good response into the HPLC or the GC is obvious, they will give the very good response. So also understand whether these extraneous peak are not belong to the solvents used during manufacturing of this particular product. The 13th point is inject individual excipient in case placebo is not available or the integrity of placebo is questionable. So understand whether the all excipients used exactly into the manufacturing of the finished product, hmm, are they not the reason for the extraneous peak. So I think these 13 different points are going to help you out in understanding the root cause behind the extraneous peak observed. So once you identify the extraneous peak's origin, the next important step can be the identification of the structure of the extraneous peak. If it is not a laboratory error, if it is a lab error, then you can definitely stop the investigation there itself and have the suitable corrective actions and preventive actions at the place. But in case if that is not the reason and if it is really something from the product, then you need to identify the structure of the peak, maybe with the help of NMR and the mass spectroscopy. Then perform the review of the available literature for that identified impurities. Now, what is the kind of impurity structure? Is there any available information? So those information details can be gathered. The third important point is now conduct the structure-based assessment. See, now you need to understand what is the toxicity of this impurity. Is it the mutagenic impurities or a non-mutagenic impurities? And for that, you need to conduct the structure-based assessment. So in this case, if the impurities observed is found to be a non-mutagenic, right? A negative result means it is a non-mutagenic. You can think of, taking the help of ICH guideline, which is Q3A or Q3B, to understand the qualification threshold. And you can decide the limit based on to the qualification threshold given into these two important guidelines. So in case if the based on to the structural assessment it looks to be a mutagenic impurities, that means your result is positive, then it can be a possible mutagenic. You need to think of conducting the non-clinical studies. Non-clinical studies, for example, Ames test or the chromosomal damage assay test. So in this test you will understand whether the substance under investigation is a mutagenic or a non-mutagenic. So in case if the negative test comes, means that your substance is a non-mutagenic, and in case if it is a non-mutagenic, you can take support of ICH guideline Q3A for API or Q3B for drug product to decide on to the limit or the qualification threshold. But in case if the substance is, if the test is positive, that indicates that now this is the possible mutagenic substance and you have to go for the clinical study. So in the clinical studies the outcome can be two. It can be a negative test or it can be positive test, because AIMS and chromosomal damage assay are in vitro test, so there can be a possibility of false positive result. So you need to confirm the mutagenicity of the substance based on to the clinical study like rodent toxicity. So in that case, if the test comes to be a negative, then this becomes the non-mutagenic impurity, and you can go back and follow the guidance given in the ICH guideline Q3A for API and Q3B for drug product. But in case if the test comes out to be a positive, now this is the possible human mutagenic impurities. So in that case, you have to evaluate the TD50. You have to evaluate the TD50, and based on to the TD50 value, you can decide the limit or the acceptable intake. You can decide for the acceptable intake. So this is the entire process you have to think of, in case of qualifying the impurities: identification followed by qualification or deciding the limit for the impurities. So based on to this entire data, you can decide the limit for the newly identified impurity, that is Q3A, Q3B, M7, nitrosamine, or the recently the recent ICH guideline Q3, which is still under the recommendation. This kind of guidelines will help you in deciding the limit for the impurities. So once you identify the limit, now the next important point is the batch disposition. So what is the root cause identified? If it is a lab error, no doubt you can certainly take the appropriate corrective action and batch can be released. But in case if it is a product related concern, then is it a cross contamination? Is it a cross contamination that is found, that is happened during the manufacturing of the batch? Then you can think about the cross contamination because of the previous product now. And what is the MACO of the previous product? What is the maximum allowable carryover? Maximum allowable carryover of the previous product. So there can be two scenarios. If your product meets the macro value, you can certainly release the batch with the appropriate kappa. But in case if the carryover observed into the product is more than the macro, then probably you have to think of, hmm, you have to think of, let me check where I was. Okay, you have to think of disposition of the batch with the appropriate corrective action, because the content of previous product is more than the macro and you will not be able to release the batch, but you have to dispose the batch, and you need to again rethink on your applied cleaning procedure. You need to think about the cleaning validation which was conducted in the past. So review of cleaning validation becomes very important in case of the cross contamination due to the carryover of the previous product. But in case if it is not a carryover, right? So the impurity is related to what? The actual drug product. So it can come from the degradation of the drug or substance, or it can be from the leachable out of the packaging materials, or can be from the excipient to packaging material interaction. So what is the available option? So you need to develop a suitable method now. See, because this current method may not be suitable, so you need to suitable a method for the observed impurities if required, and suitably validation needs to be conducted. So once you do the validation, then probably you will have the observed impurities below qualification threshold, or that impurity can be below acceptable intake in case of mutagenic impurities. So in case if it is below qualification threshold or acceptable intake, then you can release the batch with the appropriate kappa. You need to give the kappa for the observed impurities and investigate the impact on previous batches. So it is very important to define the scope of your investigation. You cannot just stop the investigation by releasing the batch, but you need to also investigate what is the response of this so-called extraneous peak, which is now known and identified into all previous batches. So those details are very, very important. Then devise the control strategy. uh, let me go back to the presentation. Then devise the control strategy to avoid or reduce the further level of impurities into the next batches. So is it possible to further minimize? Hmm? The the content of newly identified impurities. If the answer is yes, probably you have to revise your manufacturing processes. So the next point is now, if the impurity is above qualification threshold given in the ICH guideline Q3A or Q3B, or if it is above acceptable intake given into the guideline M7 or for the guideline given for nitrosamine. So this is going to be a very tough situation, and you need to dispose the batch if no reduction below qualification threshold or acceptable intake is possible for current batch in case of API. But in case of finished product, you may not be reproduce you may not be reprocess the batch once again, and the disposition is mandatory. But in case of API, you can think of whether there is any process of adopting the a purification step where these impurities can be further reduced below the qualification threshold or acceptable intake. Investigate the impact on previous batches. So you need to investigate the impact. What is the impact onto the all previous batches? Whether this impurity was observed but not noticed by the analyst. So those investigation details are very, very important. And the third point is now devise the control strategy to completely avoid the formation of this impurity or further reduce below the qualification threshold or acceptable intake. It is very important to understand what is the control strategy I must adopt so that this impurity can completely avoid it, or at least if not completely avoided, can be reduced below the qualification threshold. So these are the very important steps one has to follow in case if the impurity is because of the drug product. So I hope now these five important point must have given you an overview, right? What is the extraneous peak? What are the expected sources? How to conduct the investigation? Then how to identify impurities? How to qualify the extraneous peak? And then batch disposition. So I think this is very important point of discussion in case if you come across the extraneous peak into a chromatographic analysis. So thank you very much for watching this video, and I will meet you soon with such kind of useful and informative videos. Till then, take care and bye bye. See you soon.